Delighted to be here today. Thanks for having me. So I'm a co-founder of DZD Diagnostics. We're a small startup trying to modernize the way infectious diseases are diagnosed and treated. So antibiotic resistance is hardly a topic that needs introduction in this crowd. We all know that it's a growing crisis. Every time a new antibiotic is introduced, we could quickly see resistance to the antibiotic grow. So for example, fluoroquinolone resistance in E. coli. And this is being recognized as a global health problem. Estimates for how big of a problem this is vary, but by some projections, if these trends continue, there will be 10 million deaths per year in the year 2050. So with the rise of antibiotic resistance and it being so widespread, you would think that it would be crazy to try to treat a bacterial infection before you know what drugs that infection is resistant to, right? That would be like throwing darts blindfolded. But in fact, that's how all infections are treated today. And that's because hospitals use an old and antiquated technique to diagnose these infections called culture, or bacterial growth assays. Now, culture works pretty well. It was invented over 100 years ago by good old Louis Pasteur. And the major problem with it is that it's just quite slow. So it takes about a day to figure out what the species of an infection is, and then more importantly, to figure out what antibiotics the infection is resistant to takes another few days. So between day 0 up until day 5, a physician doesn't know what to treat their patient with. This is probably a problem that many in this room are quite familiar with. So what can we do about this? Well, actually, before I get to that, I'll say that one of the big problems of this kind of lag in time is that for many patients, they just can't wait. So we heard today a patient story of a severe infection. We've been hearing a lot about sepsis. In cases of severe infections like septic shock, the risk of a patient dying increases by 8% for every hour, not every day, but every hour that they don't get the right antibiotic. So patients can't wait 5 days to get this diagnosis. In the meantime, physicians, what do? They treat with broad spectrum antibiotics. So these don't target a specific infection, but just really carpet bomb whatever might be there. And this, of course, has its own negative consequences. These can wipe out the healthy microbiome. The drugs themselves can be toxic to end organs like livers and kidneys. You have increased lengths of stay, typically in the ICU, which is quite expensive. These overuse of these antibiotics themselves of the broad spectrums are increasing antibiotic resistance, and this leads to a pretty terrible feedback cycle where now we don't even know if broad spectrums are always going to be effective. Okay, so what can we do about the state that we're in? There's a pretty high unmet clinical need, and so there's kind of been some recognition in terms of some of the technologies that are coming out on the market. And so kind of the way I would classify some of the diagnostics that have come out or are emerging are kind of on these two axes. So there are some new automated culture techniques. So for example, the Vitek, if you're familiar with that, these are assays that fundamentally still rely on culture, so they're still growing bacteria, but they're doing it in an automated way, requiring less material. Maybe they're using robots, doing it faster. And those can be much faster than the 5-day turnaround, maybe hitting 2 days, but they're still fundamentally limited by time, right? Bacteria just grow slowly. But on the other hand, they can be quite comprehensive, being able to assess any infection that walks in the door. Now, there's a whole new class of antibiotics that are sensor-based. They use molecular probes in order to figure out what's in a sample. And these can be extremely fast, right? They can be on the order of hours. The downside with these is that they have a handful of probes that they use. So are you in these my top 5 favorite species, E. coli, Staph aureus, Klebsiella, et cetera. So you basically have hypothesis-driven diagnostic. And if your hypothesis is wrong, if that bug or if that gene is not in that sample, you're back to square one. So of course, we want to get up to this top right-hand corner here. We want to get out of this trade-off because it seems that a lot of the technologies that we have today are either comprehensive but slow or rapid but narrow. So what can we put in this top-hand corner? Well, it'll come as no surprise given what we're doing that we think a culture-free whole genome sequencing assay will be the way to get rid of this trade-off. So why is that? Culture-free will get rid of the time aspect. If we don't have to wait for bugs to grow, we can do things more quickly. On the other hand, whole genome sequencing, if you do that in an agnostic way, any pathogen that has a genome, which to my knowledge is all bacteria, should be able to be assessed with this kind of assay. Now, this is hardly a new or novel idea. Folks have been talking about using bacterial genome sequencing for diagnostic purposes for a long time, at least since the early 90s. I'm sure probably before then, far too many papers and luminaries in the field to cite. So one question is, why has this not happened yet? Why do we not see this in the clinics? If you go into the clinic today, nobody's doing routine whole genome sequencing of bacterial pathogens. So what stopped these technologies from actually entering the hospital? So there's a couple answers for that. There's a lot of kind of requirements in order to make this useful. One is the problem of time. So like we said, these patients really are degrading, their health is degrading by the hour. And so you really need something that has a quick turnaround. It's not like some of the applications, for example, in oncology, where you send out a sample, you get the diagnostic a week later. The patient's health hasn't kind of degraded in that 1 week time. Here you need something that works in hours, not in days or weeks. A very kind of difficult problem technically is that there's minuscule amounts of pathogen DNA within these clinical samples. So actually being able to access that is extremely challenging. We need to be able to do rapid and low-cost sequencing. That's not something that's been available 30 years ago, and it's only now starting to be kind of on the market. And finally, you need accurate algorithms to actually interpret the genomic data. So these are kind of all the challenges that have made it difficult to get something in the clinic thus far. I'll tell you a little about what we're doing at day 0 to try to address these challenges. We're still early stage, so ours is also not in the clinic yet either, but we think we're on the path to get there. So the way that our technology works is we take a clinical sample, and we're really focusing first on blood, on bacteremic samples, because this is kind of the area of highest unmet clinical need, where patients' health is really in a bad state when they don't know what antibiotics to give the patient. So we take a clinical sample, and the first thing that we do is we isolate the pathogen DNA using a technique we developed called blood to back. Once we have that minuscule amount of pathogen DNA, we then sequence it using a rapid sequencer. Those genomic results are then passed to an algorithm called Genome. This is a machine learning algorithm, which then spits out the two things that a physician really wants to know. What is it? What species is it? And what's the antibiotic resistance profile? And this algorithm, Genome, is built off of a training data set that we've built called MicromeDB, which allows it to learn how to do these predictions. So I'll give you a little bit of a flavor of each of these kind of sections. So when I talk about extracting pathogen DNA from blood, it turns out that it is the proverbial needle in a haystack problem. And in fact, quite a large haystack if you kind of do the back of the envelope calculation. The amount of human DNA to bacterial DNA in a bacteremic sample is about 1 billion to 1. That's a big number. And that DNA is coming from all sorts of sources, whole blood, white blood cells, but also red blood cells, platelets, cell-free DNA. So there's a lot of human DNA that you somehow have to get rid of if you ever want to see your bacterial DNA. So there's a couple ways that you could do this. Of course, one way to do it is to have some kind of a magnet which will pull out the needle, right? And this would be kind of what the PCR-based probes are doing. They're trying to ignore kind of the human DNA and just pull out what they know to be in there. But unfortunately for bacteria, there is no one universal magnet, right? There is no one way to pull out in an agnostic way. And so what our approach has been, and it almost seems nonsensical, but instead of using a magnet, we're trying to get rid of all that hay to access the bacterial that's underneath it. And so we've developed this method which starts out with a lot of human DNA in the micrograms, passes it through multiple steps, and at the end we end up with almost undetectable amounts of human DNA, while at the same time retaining our bacterial DNA, and we do that in an agnostic way. And when we look at the sequencing from this process, we can see that the majority of our reads, the majority of our sequencing reads, are actually the bacterial pathogen. And these are experiments that we've done where we've spiked in just one CFU, so one bacterium in a milliliter of blood, 1 CFU per mil, and we've been able to get great results on the sequencing side. So speaking of sequencing, how are we actually doing that? Well, I'll say one of the barriers to actually doing this in the past has been the cost of sequencing. So back in the early 2000s, when the Human Genome Project was completed, it cost about $10,000 to sequence a megabase of DNA. Nobody was going to use that for a diagnostic for pathogens. Fast forward to today, it's now costing cents per megabase. So really cost isn't the barrier anymore for pathogens. And I'll just kind of mention, since a lot of the talks here have been human genome based, a bacterial genome is about 1,000 times smaller than a human genome. So what you can do for humans, you can do 1,000 times cheaper for pathogens, bacterial pathogens. And kind of secondly, there's all these new technologies that are now coming out in the market. Some people are calling them third-generation sequencing technologies, which really enable rapid diagnostics. So a notable example of that is Oxford Nanopore Technologies with their MinION device. We make a lot of use of it. It has the great advantage of not only being small and portable, but also extremely rapid. You can do all the sequencing that you need to do for a bacterial pathogen in about an hour. So we're not actually doing any innovation on this front. We're leveraging the technologies that already exist in the industry. Once we sequence our bacteria, we then have to do analysis on it. The first thing we do is species ID. Now, there's actually plenty of really wonderful open source algorithms that can do species ID based on genomic sequence. So I won't really talk about too much there what we do. But I'll just note that the ability to use whole genome sequencing for pathogen ID really enables you to open up to comprehensiveness in terms of all the infections that can occur. So if you look at the abundance of pathogens that occur in blood infections and bacteremic samples, you'll see that kind of at the top of the list are our usual suspects, things like Staph aureus, E. coli, Klebsiella. But then you start to see that it's a very, very long tail. And you get down to this right-hand side, and there's some bugs there that I usually hesitate to try to pronounce in the presence of real microbiologists, right? It can get quite weird. And the point is, if you use a PCR-based assay that maybe hits your top 5, you're missing a large part of this tail. And so whole genome sequencing, of course, it has to be validated for each bug, but it has the potential to really hit this whole spectrum. So it's a very powerful methodology. The next step that we have to do is actually predict antibiotic resistance, because we want to know what to treat the patient with. Now, the traditional way of doing this is to have a lookup table of resistance genes, resistant mutations, and then try to see if your pathogen has those resistance genes. And this can work extremely well. So for example, for Staph aureus, methicillin resistance is mediated by a single gene, MEK-A. And if you look at a cohort of samples, if you look at the ones that are resistant, almost all of them have the gene. If you look at the ones that are sensitive, almost none of them have the gene. And so it's a perfect predictor for antibiotic resistance and this particular combination of species and drug. But that's not always the case, right? And so there's plenty of cases, for example, Cefazolin resistance and K pneumonia. If you try to do the same thing and try to use whatever genes you can find in your database, you'll find that you're not able to predict all the way, right? There's plenty of unknown resistance that doesn't allow you to do a robust job telling a physician, okay, this is the drug that it's resistant to. And so the way that we think about using these traditional databases, we think they're highly interpretable and they're backed by scientific understanding, which is very good for explanatory and being able to understand how the algorithm is working. But on the other hand, they are quite limited in terms of what's already known in the scientific literature, and can be a little bit rigid in terms of more complex interactions. So if you have epistatic interactions, if you have multiple genes interacting, it's hard to capture that in this kind of traditional approach. So our approach has been to take a more modern machine learning approach where we use a large data set in order to learn how to do these predictions. This allows us to have a flexible model. It can be agnostic and can learn new resistance mechanisms. Of course, the downside of that is that it's more difficult to be interpretable, right? This is kind of what people always talk about, the black box of machine learning. We try to get around that by having thoughtful integration of biological knowledge into our model, but it's something that we're continuing to work on. And kind of the nice thing about it is that it scales with data. The way I kind of think about this is moving away from the GWAS or the single SNP association to more kind of a more modern method is similar to the evolution that we've seen in many other areas of machine learning. So for example, face recognition technologies, there's these amazing papers from the 70s where in order to recognize if this face came from this particular person, they would measure kind of the distance between the eyes, the width of the nose, et cetera, and they would try to do a matching. So really handcrafted features. Now, fast forward to how Facebook is doing things, you can imagine that they're not doing any handcrafted features and measuring of facial dimensions, but rather they're using an algorithm which can learn those features from the data itself. So that's the approach that we're taking. And of course, in order to do that, you need a large data set in order to be able to actually learn those features. So in order to actually develop that data, we have been creating a large database called MicromeDB. We've been collecting many clinical isolates, sequencing their genomes, and entering them into our data set, which consists of the genomic sequences of the pathogens match their antibiotic resistance pattern. And so our algorithm, Kenome, is able to learn from this data set such that when it sees a new genomic sequence, which it's never seen before, it tries to predict its antibiotic resistance pattern. Like any good machine learning algorithm, it gets better with the more data you give it. There's a lot of plots on here, but x-axis here is the number of samples the algorithm was fed, and the y-axis is accuracy, and all the plots go up and to the right, which is exactly what you want to see. Things get better with more data. Kind of interestingly, though, it really depends on the combination of species and drug. So for some cases, like Staph aureus fluoroquinolone resistance, it's quite easy for any algorithm to learn that from very, very few examples. But if you move to other types of resistance, like clindamycin resistance in Staph aureus, then you start to see that you actually need more data in order to do a robust job predicting. So we're at a point where we have over 30 of these models working pretty well, over 95% sensitivity and specificity, but we're constantly working to try to improve both our predictions and the number of combinations that we can do accurately. So finally, I'll say, so we have kind of a lab-based protocol. We're working to develop that into a prototype and into a device that will eventually sit in the hospital clinical microbiology lab. And the vision is that once that's there, once we have these devices in hospitals actually diagnosing infections, that becomes a really powerful tool. Because every time our diagnostic gets used, it will generate a genomic data point. And that genomic data point will get sent and added into MicromeDB, into this data set of pathogens. And that data set starts to enable lots of new opportunities. So things like large-scale epidemiological studies, hospital outbreak investigations, antibiotic target discovery, and those will all be kind of fed back to the diagnostic and to improve services to hospitals. So briefly, I'll talk very quickly about some of the work that we're doing in terms of hospital outbreak investigation. Hospital-acquired infections are, again, kind of a well-known problem. It affects a large proportion of patients in the United States. CDC estimates that about 100,000 patients die every year from hospital-acquired infections. Now, we heard in an earlier talk from Jennifer's work, whole genome sequencing is a phenomenal way to figure out if a transmission event occurred, right? You get down to very, very high resolution to see, are these two bugs related or not? Unfortunately, we don't see widespread use of whole genome sequencing for outbreak detection. We don't see it in infection control. And the reason for that is infection control teams don't have access to these types of technologies, to these types of analyses. And so they're stuck using epidemiological assays. They're looking at lab reports. They're using maybe some older technologies like PFGE and MLST, none of which have high resolution or really great for telling in detail whether or not two infections are related. So this is clearly a place where whole genome sequencing could be an enabling opportunity, both in a retrospective and in a prospective manner. So just kind of as a pilot service, what we've been doing is offering to MGH and the Brigham kind of a research service. Well, they'll send us isolates which they suspect to be related in some kind of a transmission event. Maybe there's a MRSA outbreak in the NICU, and they want to know, are these related or not? And we do a SNP analysis and return to them in 2 to 3 days a very clear report that says these are related, these are not related. And this is something that they can then use, and they can cross-check with their epidemiological data that they have about these patients to be able to say, okay, here's how we take this, here's how we understand, are we going to do an intervention, are we going to clean the ward, et cetera. So this is just kind of one of the ways where we think that whole genome sequencing is going to modernize the way infectious diseases are diagnosed and treated in hospitals. So with that, I'd like to thank my team and thank you all for your attention, and happy to take questions at the panel.